Einstein once said, "The important thing is not to stop questioning. Curiosity has its own reason for existing." Curiosity and intellectual hunger led humankind to groundbreaking discoveries in science, such as radiation phenomena, studies of the structural and functional organization of the cells, optical tweezers, novel cancer therapies, gravitational waves, mechanistic studies of DNA repair, and the discovery of split genes. Such discoveries push humankind forward to a better tomorrow, with fewer terminal illnesses and a greater understanding of the world surrounding us. Welcome to the NYU Tandon School of Engineering and our latest seminar in what has proven to be an important and informative series, whose goal is to unify scientific communities around the influential research ideas of today that can have an impact tomorrow. The series aims to bring together researchers and students to discuss recent advances in artificial intelligence and related fields. AI is already recognized as an important research area that enables advances in the fields of medicine, biology, neuroscience, and more. We hope that the scientific discussions that result from this series will contribute to building a better tomorrow and promote continued groundbreaking discoveries. I would like to thank Dean Jelena Kovacevic for her utmost support, as well as my own Department of Electrical and Computer Engineering for hosting this series, which is evolving into a significant venue. When I first conceived of this series, I hoped that I would be able to attract highly esteemed speakers doing revolutionary work in their respective fields. I say with some humility that I have succeeded beyond my wildest dreams. For proof, look no further than our guest today. It is an enormous honor to welcome Nobel Prize winner Sir Richard J. Roberts to Tandon. Although his fame in scientific community precedes him, I'll take this opportunity to say a few words by way of introduction. Now the Chief Scientific Officer at New England Biolabs, Roberts won the 1993 Nobel Prize in Medicine. His impressive CV also includes a doctoral degree in organic chemistry from the University of Sheffield, postdoctoral work at Harvard, a stint as Assistant Director of Research at the Cold Spring Harbor Laboratory under the fellow Nobel laureate James Watson, and more awards, fellowships, and honorary degrees than I could ever hope to list here. I'm pleased to say that his lecture does not mark the last time a Nobel laureate will take part in the NYU Tandon ECE seminar series on modern artificial intelligence. Next semester, we will hear from Eric Kandel, university professor and Fred Kavli professor at Columbia University and a recipient of a 2000 Nobel Prize. Other confirmed speakers include Marshall Hubbard, professor at Carnegie Mellon University, Tony Jabara, professor at Columbia University and the director of machine learning at Netflix, and Manuela Veloso, Herbert A. Simon University professor at Carnegie Mellon University and the head of JP Morgan AI Research. Now, without further ado, let me invite Sir Richard J. Roberts to the stage. Richard discovered split genes and the mechanism of RNA splicing. That research resulted in a fundamental shift in our understanding of genetics and has led to the discovery of split genes in higher organisms, including human beings. He demonstrated how RNA can be divided up into introns and exons, after which the exons can be joined together. That process can occur in different ways, giving gene the potential to form a number of different proteins. With a keen interest in sequencing DNA, Richard also became heavily involved in using computers and was a pioneer in what is now called bioinformatics. Ladies and gentlemen, hats off to our speaker. I give you Nobel Prize winner, Sir Richard J. Roberts.
Okay, well, thank you. It's a great pleasure to be here. I'm going to give a fairly high-level talk about my life in science. I will introduce on a couple of occasions some areas where I think AI and machine learning can play a major role going forward. And I would have you know that our company is actually trying to hire someone to start up a small AI group to look at this and to look at other problems that we have within the company. I would just draw your attention to this little picture here. This is a young lady hugging a bacterium. I work with bacteria. I love bacteria. You too should love bacteria because they are keeping you alive. Almost all of the bacteria that live with you are making sure that you have a healthy existence. I was born in 1943 during the Second World War. I was fortunate. I lived on a street that was in the direct line of the German aircraft trying to bomb the Rolls-Royce engine factory. They were making all of the engines for the RAF at that time. Half of the street was destroyed, but I was lucky. The half that I lived on was not destroyed. And this will be a recurring theme throughout this talk, just how important luck is, and I would have you all think about this as we go through the talk, and think about your own lives and where luck has played an important part, because it's really quite amazing what it can do for you. So the town I was born in is in the middle of the country. It's called Derby. It's labeled with a little red arrow at the top. And then I moved when I was 4 down to the city of Bath. If any of you go to England and are looking for somewhere good to go and do a little tourism, I would absolutely recommend Bath. It's a beautiful city. It's 2 hours on the train from London, and a wonderful day can be spent walking around looking at the architecture. It really is quite an extraordinary city. I grew up in Bath and stayed there until I went to university. This was a small school that I went to. And this school I went to when I was 7, from age 7 to 11. And I show this because the headmaster at this school had an amazing effect on me. For some reason, when I would leave in the afternoon to go home, he would give me a little puzzle, usually a mathematical puzzle, to do. And as a result of that, I fell in love with mathematics, to a point where if you'd asked me when I was maybe 15 or 16 and what I really wanted to do with my life, I thought mathematics was going to be my future. However, that changed a little bit, because when I was 11, I'd read this book about how to become a detective, and that seemed pretty good too. And then my father bought me a chemistry set for Christmas, and it was one of these chemistry sets, not like the ones you buy now where all the interesting experiments are not possible. This was one I discovered I could make fireworks, I could make explosives, just so much fun to be had out of this chemistry set. And that sort of led me down the path of wanting to be a chemist. My father built what was a makeshift lab in our kitchen. We actually only had one room that we lived in, so it was the kitchen. But my parents were not educated. They both left school at age 14. They had no idea what I was doing, but they saw that I loved what I was doing, and so they were tremendously supportive throughout my life. They've always been as helpful as they possibly can, because they saw I was having fun, and fun is something that's very important in life. Now, when I was 11, I went to the City of Bath Boys' School, which was the local grammar school in Bath, and this was the school that people would go to if they were going to sort of go into a higher level of education, if you were going to go to university later on, or you were going to have a profession, as opposed to the technical colleges where you went if you were going to be a plumber or a carpenter or something like that. What happened in Bath, the City of Bath Boys' School, I increased my love of mathematics. I learned to play the violin, which I enjoyed for quite a long time. I unfortunately don't have the time anymore because I'm not a very good violinist, and it takes me a lot of practice even to make the thing sound decent. I also got interested in caving. There was a limestone plateau very close to Bath. Used to go down caves, and I loved the fact that you could go down, and every so often you would go into some part of a cave where no one had ever been before. And so this sort of fostered my love of exploration. And now, of course, I do it in different fields, but this really got me very interested in wanting to be the first to do something. And so that was quite good. And Bath was also famous for jazz. Now, I went to Sheffield University. I went there because I decided I was going to do chemistry at university. There were two good universities. One was Southampton, one was Sheffield. I applied to both. Southampton didn't take me. Sheffield did, so I went to Sheffield. I got a BSc with what they call a 2-1. It's not a first, it's not the top, it's down a little bit. And it's interesting that I've noticed throughout my life that very often it is the people who are in the second tier, very often, who are the most successful in life. I don't know why this is, but I do know that the 6 people who all got a first-class degree in my class actually have sort of mediocre jobs and have not had terribly interesting lives, at least not by my standards. So when I take students, when I bring people in to work with me, I don't care if they've been really good academically. I want to know, are these people who are enthusiastic about life? Do they really want to do what is fun? Or are they just trying to tell you how good they are because of their book results? So I'm a little skeptical of those, I must admit. So anyway, I got my bachelor's and stayed on and did a PhD in chemistry with a fellow called David Ollis. He was professor of chemistry. He's one of the best chemistry teachers that I ever had, but that was at the undergraduate level. When it came to go to his lab, he put me in a lab with a postdoc, and I rarely saw him. I would see him occasionally. But this postdoc was the best mentor that I've ever had. So after the headmaster who got me interested in math, this fellow is featured there, Kazu Kurosawa. He's a Japanese gentleman, and he was a postdoc in the lab, and he taught me more about chemistry and how to do chemistry. You know, one of the things you're doing research, you have to know how to do it, and you have to know what sort of questions to ask. You've got to know how to go about answering them. And he was one of these rare people who would suggest, well, I think it would be a good idea if you did this, and he would explain it so well that at the end of it, you knew exactly why you had tackled the problem that way. And he was just fantastic. And a couple of years ago, I met him again. We had communicated for a little while after he left Sheffield and went back to Japan. We used to play Go by mail for about 2 years, 3 years, until his English got so bad it was difficult for him to write a letter to accompany the moves. But I met him a couple of years ago with his family and his grandchildren, and we had a grand old time in Tokyo. But during this time, after Kazu left, I had all the results I needed for my PhD in the first year because of Kazu. And, you know, but you can't write a thesis after a year in England. You had to spend 3 years to do your PhD. And so I sort of just fooled around in the lab, I would say, for about a year. And I used to go over to the library and I would read about stuff. And I came across this book called The Thread of Life by John Kendrew. And I'd already read a little bit and heard a little bit about this subject they call molecular biology. But this book was, for me, just wonderful. It described the origins of molecular biology and what people were trying to do and what was interesting. And by the time I finished the book, I knew this was what I wanted to do. And I've often found during my life that I will read a book outside of the subject I'm working in, and it sounds so interesting that I want to do that. And I've never felt a problem in sort of switching fields. If you see something you really want to do, it's actually quite easy to switch. You know, I moved from math to chemistry, from chemistry to biology, to molecular biology, and then into bioinformatics, which is what I mainly do at the moment. So this book, I would recommend it. I wrote in my Nobel autobiography that it had had this effect on me. And John Kendrew himself wrote to me, and he said this was the nicest thing that anybody had said about his book, because apparently the critics panned it and said it was awful. But for me, this was the thing that made the difference. So when it came time to do a postdoc, I wanted to switch fields from organic chemistry into molecular biology. I applied to 6 labs, 5 of them turned me down, and this one gentleman, Jack Strominger, who was at Harvard, took me up. He offered me a position. And he at the time was in Wisconsin, and I thought I was going to go and be a postdoc in Wisconsin at Madison. But he wrote to me about 2 months before I was scheduled to move, and he said, can you just delay coming for a couple of months, because I'm moving to Harvard, and you have to come to Harvard. So that's what I did. I picked up, went to Harvard, and worked with Jack. Now, Jack, I had heard him speak at a meeting in Sheffield, And he described about 50 man-years of work in 1 hour and made it all sound very easy. So I thought this sounded good. When I got to Harvard, he had decided to change fields. He wanted to do something new. He'd worked previously on bacterial cell wall biosynthesis, so he was really a bacteriologist, which is why we have the icon at the bottom right-hand corner. I love bacteria. But he had a problem that he was waiting to finish up about the role of transfer RNAs. These are small RNAs that carry amino acids around in cells, that worked in bacterial cell wall biosynthesis. So bacteria put short strings of amino acids into their cell walls. And this particular tRNA doesn't work in protein synthesis. It only works in bacterial cell wall synthesis. And so he gave me this project to work on. There was a previous postdoc who had been doing it, and he had an idea about how to do it, but was rather an old-fashioned idea, but I knew nothing. So I read the literature, looked around, and discovered that there was a gentleman called Fred Sanger who worked at the MRC lab in Cambridge who had developed methods for sequencing RNA. He actually previously won a Nobel Prize already for developing methods for sequencing proteins. And so I wrote to him and said, could I come and learn how to do it? And he said, sure. I went. I spent 6 weeks in his lab, writing down everything that I possibly could, went back to Harvard, set up a replica, mini replica of his lab, and everybody then came to me to learn how to do it. And so I met a load of people. It turned out by chance I was the first person in the Boston area to doing this. So I met a lot of people that way. And Fred was really my hero in many ways. He was one of these people. He was very modest, would never tell you that he was good or anything else. And if he made a suggestion to you, it was always one, well, maybe you could try this and see if something could happen. I met him. I went into the lab on a Sunday morning after I arrived, and I found this gentleman who was busy washing up some labware. And I said, hello, I'm Rich Roberts. I've come to work with Fred Sanger. And he said, oh, hello, I'm Fred Sanger. So that was how we met. Now, during the time at Harvard, I did end up sequencing some tRNAs. And just as I was about to leave, I wanted to come back to the UK because, I sort of have British blood circulating around in here. Went back and applied for a job in Edinburgh. And I'd been told that there was this wonderful job waiting for me in Edinburgh, and I was the perfect candidate for it. And so I sent in a letter and never heard anything. Just nothing came back. In the meantime, I got offered a couple of jobs in the US, one at Harvard and as an assistant professor, and one down at Cold Spring Harbor with Jim Watson. And that was a very strange job. Mark Batashny came up to me. He's a professor. He's now down here in New York, but he was at Harvard at the time, and said, well, Jim Watson has the perfect job for you, and he's going to come and talk to you about it. A couple of weeks went by, and nothing from Jim. And so Mark came up to me and said, well, Jim doesn't actually know who you are, so perhaps you could go and introduce yourself. So I go, knock on his office door, go into the office. Within 5 minutes, he'd offered me a job. 5 minutes later, he made it clear he didn't want me to hang around anymore, and so I started to leave. And he said, oh, and by the way, you probably should come down to Cold Spring Harbor and see what we're all about down there. But you've got the job. Don't worry, he said, you've got the job. So I ended up going to Cold Spring Harbor. Now, that turned out to be interesting. The reason he wanted me to go to Cold Spring Harbor was to sequence a small virus called SV40, which was a very small virus that could cause cancer in human cells and in monkey cells and various others. But when I got to Cold Spring Harbor, I discovered there were 2 other groups already sequencing it, not at Cold Spring Harbor, elsewhere. And I'm not a big fan of competition just for the sake of competition. I much prefer collaboration. But what had happened is just before I went down, I'd gone to a seminar at Harvard given by the man in the top left there, Dan Nathans. And he had worked on SV40 DNA, but using something called a restriction enzyme. This is an enzyme that Ham Smith in the bottom left had discovered that will take piece of DNA and cut it into fragments at very distinct sequences. So you could take the 3,500 bases of SV40 and cut it into about 8 or 9 fragments. And some of these were quite small. Now, one of the reasons that RNA sequencing was developed before DNA sequencing is because if you're ever going to develop a new method, you need something simple to practice on. And especially for sequencing, you can't start saying, I'm going to work out how to sequence 3,000 base pairs. You have to start with something that's 50 or 100 or nice and short so you can develop the methodology, make sure it works, and then apply it to longer things. And it occurred to me that this was the way to make the small pieces of DNA upon which one could learn how to sequence. And I, in my naivete, thought that I could do this, and I would develop DNA sequencing methods and beat Fred Sanger, because I happened to know he was interested in this same problem. Well, to cut a long story short, our attempts to use these to sequence DNA were lost because we started to look for more restriction enzymes, and everywhere we looked, we found them. And these enzymes, which would cut DNA into well-defined pieces, turned out to be the basis of the biotech industry. And so one of the reasons that we have a good biotech industry today is cloning, but cloning needed restriction enzymes. And these were really the workhorses of the biotech revolution. And so we'd found 75 in my lab, 75 of the first 100 that were known, and everybody wanted them. People would come to meetings and they'd bring DNA and they'd sneak out of the meeting and ask, did I have something that would cut up their DNA? And so I suggested to Jim Watson that we should start a company at Cold Spring Harbor. We should offsite, but we would sell these enzymes to the biological community and use the profits to support the research at Cold Spring Harbor. Now, Jim Watson, in his wisdom, said, well, he's not interested in that. Didn't think I could make any money doing it. It was very dirty to be associated with industry. This was back in 1974. And he didn't want anything to do with it. In fact, he actively discouraged me from doing anything about it. So because we were giving away all these enzymes and we ended up, it was clear there was a need for it. And so I looked for alternative sources. Eventually ran into a guy in Boston called Don Combe, and that led to the formation of New England Biolabs, and I'll tell you a little more about that later on. Now, Jim Watson is not the easiest person in the world to deal with, and since he'd hired me to sequence SV40, and I made it pretty clear I wasn't going to do that from the very start, he used to get mad at me all the time. And this is why I mentioned Norton Zinder. Norton Zinder was here at Rockefeller University and probably one of Jim Watson's very few really good friends. But Norton became very interested in myself also because of the restriction enzymes, and we became good friends. And Norton was the person who, when Jim said, well, I'm going to fire you, Norton would go to Jim and say, well, no, why don't you keep him on, give him another chance? And this was continual, so I would say for almost my entire time at Cold Spring Harbor, I fought with Jim Watson. Now, Cold Spring Harbor. One of the things that happened after we spent a lot of time looking for and characterizing new restriction enzymes, but we wanted to do something with them. We wanted to find some interesting biology to get involved with. And so we got involved in a project to look at promoters. These are the sequences in DNA that tell the RNA polymerase where to start making the messenger RNA so that a gene can have its information transferred into protein. And at the time, there were several groups at Harvard and one in the Pasteur who'd been looking at this in bacteria to see what these signals were that said start making RNA, but no one had done it in eukaryotes. And so we thought it would be good to do that, and we started studying adenovirus. And my postdoc, Richard Gelinas, who I had known actually as a graduate student when I was a postdoc at Harvard, came down and joined me and started on a project. And the idea was actually a fairly simple one. The idea was we would look, well, I think maybe I kept this next slide. Yeah. So messenger RNA is interesting. It's got, at its 5' end, this funny cap structure, a triphosphate and an inverted guanosine and so on, and then at the 3' end has a long poly-A tail. And there's an enzyme called ribonuclease T1 that will cut RNA every time there's a G. And in this way, it gives 3 kinds of oligonucleotides. One that contains this cap structure at the 5' end, one that contains the poly-A at the 3' end, and then all of these others in between that are just, they have a 5' hydroxyl, 3' phosphate. And so I worked out an easy way to separate these, and Richard started doing the experiments. And we decided to look at adenovirus because if you look, this was what was known roughly about adenovirus, and you can see a lot of messages. But in particular, the ones labeled E, E1A, E1B, and E4, these come in from the end of the linear virus. And we thought if we can get the sequence of the RNA at the 5' ends of those RNAs, then we could just sequence in from the end of adenovirus DNA and look and see where this sequence was, and then we would know the sequence immediately preceding it must be the promoter. So we tried doing that. We couldn't get enough early RNA, eRNA. We just couldn't get enough to do the experiments we wanted to do. So we started to look at late RNAs, and those are all the things marked with an L on this slide. And you can see there are quite a lot of them. There's something like 15 or 16 we were expecting. And so we thought we'll take total late RNA, cut it with this enzyme, ribonuclease T1, and we should have 15 or 16 oligonucleotides that will tell us where these messages start. And if you think this was sort of the early days of trying to map transcripts, this was just an alternative way of trying to do it. So Richard did the experiment, and this was the result. We discovered that just shortly after infection, there were 2 spots, only 2 5' oligos. Late in infection, there was only 1. And these 2 on the left are actually a regular and a cyclic form of the one on the right. So there's really only 1 end to all of these RNAs. And it didn't make sense, We knew these things, they're starting at different points in adenovirus. And so Richard, I said, go do the experiment again. And so he went and did it a couple more times and got the same result. And so, I said, well, why don't you let me do it? I'll show you how to do this experiment properly. And so I did it, and of course, I got the same result, because if anything, Richard was maybe even a better experimentalist than I was. And so we knew, as soon as we got this result, and it was confirmed, that there was something going on in adenovirus and in eukaryotes that was different from anything we knew about in bacteria, because in bacteria you just don't get this result. And so the question was, what is it? What happens? And so we got this result in early 1976 and spent the next year or so trying to figure out what was going on. And the bottom line, to sort of cut a long story short, is everything we tried to do said that this oligonucleotide, while it was at the end of all the messenger RNAs, was not coded at the end of all the messenger RNAs. It was coded somewhere else. So this was the paper, a predominant undeconucleotide, published in July 77, submitted, I think, probably about January or so that year. And so here was the idea that we came up with. And if we look on here, the strand at the top is the RNA, and it's an exact match for the DNA in the little bit that's going to code for the protein. But at the 5' end, there's a bit sticking out, and it sticks out because it's not directly coded next to the main body of the message. And at the bottom, you can see what we call an R loop. If you take this hybrid at the top and you just heat it up a little bit with double-stranded DNA, then you can form these R loops. And that's because RNA-DNA hybrids are more stable than DNA-DNA hybrids. And so the thought was, if you could do that, then we could find a piece of DNA that matched the 5' end, match this bit sticking out at the 5' end, and then do an electron microscope experiment to look and see what it looked like. And the prediction was that it would get an R loop and there'd be stuff hanging on to it. Now, neither Richard nor I were electron microscopists, but we had a couple of really great electron microscopists just down the hall, Tom Broker and Louise Chow, shown here. And we went to them on a Saturday morning and said, we got this idea for an experiment. Can you do it for us? They did the experiment, and on Tuesday morning, the very first molecules they looked at were exactly of the form that we had predicted on the blackboard when I suggested this experiment. However, there was just a slight problem, that is, instead of hybridizing at just one point, the probe we used hybridized at 2 points. And that's because the genes were split into pieces. This discovery was published in Cell later in 1977, in September. And it has an interesting title, an amazing sequence arrangement. When we sent it out for review, the 3 reviewers all wrote back and said that it was inappropriate to use the term amazing in a scientific paper, that this was jargon, it wasn't really very scientific. And the editor, Benjamin Lewin, also in his letter said, can you find another word for amazing? And so I called up Ben, we knew one another, and started talking, and during the course of the conversation, I said, what did you think when you first saw this result? And he said, oh, it was amazing. I said, I rest my case. And so he let the word amazing into the title, which, as far as I'm aware, was the first time it's been used in the title of a scientific paper. And this is really what split genes are all about. So at the top, a gene is just a straight piece of DNA. You read through it 3 bases at a time, and then this accounts for the protein, and the messenger RNA looks exactly like it. However, in eukaryotes, the genes are split into pieces. And in order to get the contiguity of the protein coding sequence, you first have to splice the initial transcript, which we call the pre-RNA, as shown here, so that the bits that code for the protein all come together, and then it looks just like a bacterial messenger RNA. That's it. That's split genes, that's splicing. It turned out that not only did adenovirus do it, everywhere you looked, you found it. It had not been predicted, which is always nice. It is not so much fun when you just discover something that some clever person had already deduced. But as soon as we got this, everybody started looking in their notebooks, discovered they had all the evidence for it already. I remember I went to a splicing and RNA meeting at Cold Spring Harbor, and one of the speakers in my session showed a picture in which he could now interpret something which he hadn't previously been able to interpret because of split genes. So that got a lot of attention. People started talking about Nobel Prizes, but that took much longer. Now, during the period from 1978 to 92, the time following this original discovery, and we thought there were several things we could do. It would be good to sequence some of these splice junctions and see if we could work out what the code was that the cell was using, where it knew to hook the pieces together. In adenovirus, it turned out there were so many places where this was happening that we had to sequence the entire genome. And in fact, the adenovirus genome, which we sequenced, was the second longest genome ever sequenced. The first had been done by Fred Sanger in Cambridge. He did lambda DNA. This was about 36,000 bases long. But at the time, there were no programs that would take small pieces of DNA and hook them all together so that you could determine the sequence. So we had to write our own programs for that, and we really were pioneers in that field. And then, of course, you wanted to interpret the sequence and work out where the genes were and so on. And so this was when bioinformatics got started. You notice I always write it capital B-I-O, because it's really more about biology than it is about informatics, to be honest. The computer scientists who got involved in this thought it was all about informatics, but an awful lot of them never really did very much because they ignored the biology. They thought you could do everything just by computation. So this was how I got interested in bioinformatics. In '93, was lucky enough to win the Nobel Prize together with Phil Sharp. He was a guy, he'd been at Cold Spring Harbor, but he'd moved up to MIT long time ago and totally independently came across the same discovery, also using adenovirus. Now, one of the things that happens, and I should say this was 25 years ago yesterday, so yesterday was the 25th anniversary of this, so that was kind of exciting. When you go to Stockholm, you have to give a lecture, your Nobel lecture, and I'd not worked in the field forever. Most people go and they tell you about all this fantastic work they've done since they made the discovery, which is what Phil did, but I really hadn't done anything. But just before I had to go to Stockholm, we made this discovery. Working with Xiaodong Cheng, who was a postdoc at Cold Spring Harbor at the time, we discovered that for DNA methylases, enzymes that put methyl group onto DNA, the way in which they do it is they bind to the DNA and then flip out the base into a space in the enzyme where they then do the chemistry. And so I was able to use this discovery as my Nobel lecture. And of course, if you make a big discovery, it's always the Swedes that you really want to tell about it. So I was happy about that. This turns out to be a very general phenomenon. Almost all enzymes that do chemistry on DNA, on the bases of DNA, use exactly this mechanism. We think this is also how DNA polymerases and RNA polymerases begin to open up a helix when they're going to either replicate them or transcribe them. In 1974, go back, New England Biolabs was founded. It was founded by a man called Don Cohn, his wife, and they had one technician. And it was then that I called them up and said, hey, we've got all these restriction enzymes. They had already expressed an interest in selling them. And so I went up and they offered me a partnership in the company, which I didn't want. And so I said, I'll just be your chief advisor. And so I ended up as their chief advisor. What I did was to provide them with a list of all these people who'd been coming to my lab because they wanted samples. And I got a mailing list, told them which enzymes I thought people wanted, and they immediately made them and made good quality ones. And then sent out a letter, just a sheet of paper saying, if you want to buy this one, it costs you this much. Buy this one, it'll cost you this much. They made $200,000 in the first year. Right? Not bad back in 1975. So we now are a company of 365 people working in Ipswich. We have about 150 people working elsewhere. And the work in Ipswich, where we make all the products, about 100 of those people, a little over 100, are doing research. Most of it is basic research and all paid for by the profits from New England Biolabs, which is what I had in mind for Cold Spring Harbor, but Jim wasn't interested. So what have I been doing since I got to Biolabs? Well, one of the things we did is we had a big program to clone the genes for the restriction enzymes just to make production easier. But in the process, get a lot of sequence information, and so it's sort of interesting to look at the sequences and figure out how it is these enzymes do the wonderful things they do. We also started to explore what the current limits of bioinformatics were, and I'll say a little more of that in a moment. And then we did the idea that, well, maybe you could just sequence a genome and work out where the restriction enzymes were so that you don't actually have to purify the enzyme and work out its protein sequence to find the genes. Maybe with bioinformatics, we can find the genes. Now, I'm going to take a slight digression to tell you about a project called Combrex. And Combrex is still a project. It's vaguely going on at the moment. We did get some grant money to pursue it for a while, but unfortunately that ended, and we've not been able to persuade anybody else to fund it. And the idea is as follows. You want to take, say, the sequence of a genome. You want to look and find out where all the open reading frames are, make your best guess as to what the genes are actually doing, and then to make those genes and the gene products in the lab and test whether your idea was correct. So, it's ideal, make a prediction, get somebody to test it. And by making predictions and then finding a lab where they were expert in whatever your prediction was, it costs very little for them to do the extra work. It's an ideal student project just to test it and see if it works or not. And so the idea was that we would start this project, we'd build the database, get a lot of computer scientists involved in this, a lot of bioinformaticians. We had people in New York who were helping us with this. And then we would find the labs that would test the predictions. And we did this for 2 years, had a lot of success, but NIH, in their wisdom, were not interested, partly because it's a low-throughput project. You have to do it one gene at a time, and NIH liked things where you can do 1,000 genes at a time. So we couldn't persuade a study section this was worth doing. Just to give you some idea of the general problem here, if you look at, these are just some typical bacteria, Haemophilus influenza causes disease. Of the 1,600 genes, about 1,200, you can make a reasonable prediction about what you're doing, but maybe a fifth of those are wrong, maybe a little more than that. 429 totally unknown. If you look at the bottom, Pseudomonas aeruginosa is a nasty pathogen. It's all over the place. Here, 42% of the genes in this organism, we have no idea what they do. And even the 3,600 that we think we know what they do, most of them have never been tested, and we now know, just looking in GenBank and in the big databases connected with this, that a lot of these predictions are incorrect. And the reason they're incorrect is because they've never been tested. And the way the predictions were made, they look and see, well, this protein, does it look like something that we already know what it does? If it does, then it picks up the same name and says this must be the function. But then someone looks at another one and finds that this one that we made the prediction of is the closest match. And so then the annotation gets transferred. And before you know where you are, you're labeling things that are 10% similar to something that you really know the function of and giving it that same function. And most of the time you're wrong. So that's a big problem. So why has there been so little progress here? Well, part of it is the inherent difficulty. It's not always that easy to make good predictions. And I think this is an area where AI, machine learning, can certainly help by looking at some of the enormous sequence data sets at the moment and just seeing what is going on. So for instance, if you look at 10,000 pseudomonads and you discover they all have this same gene, but we don't know what it is, but it's perfectly conserved all the way through, that tells you it's probably an important gene. Very often you can have a hint as to what it might be doing and then go and do the functional test. So there's a lot of stuff that can be done. You need a cross-disciplinary set. You need people who are good on the AI front, good on the computational front, but also good biologists and good biochemists who can do the biochemistry to test the predictions you make. The appeal of doing everything on the whole genome and making predictions has just taken over. So most people who are working on the human microbiome, on all the bacteria that live with you, they're content just to sequence them and then make some guesses and then move on to the next project. And people have not been terribly focused on looking at individual genes to work out what they do. Another major problem is the lack of what I call the gold standard protein database. That is a database in which all of the proteins that we know the function, because it's been experimentally determined, exist. This database doesn't exist. I don't understand why. We have one for my field, for restriction enzymes, because we put it together, but no one else has such a gold standard database, and that's definitely needed. And then a lack of appropriate funding mechanism. So NIH, NSF, the traditional methods just don't work. They're not very good for this. So now I want to tell you about something that I'm doing at the moment, and this is a way of finding out where methyl groups are in bacterial DNA. There was a method developed by Pacific Biosciences for sequencing DNA that gives you two things. It gives you very long sequence reads, but it can also tell you where the methylated bases are. And it depends on this. As the polymerase is going along, the DNA polymerase is copying a single molecule. If the template has a modified base, the polymerase slows down. And so you can measure how long it takes to add each base. And because it slows down, you know there must be modified base in the template. And it turns out this works really well. We spent quite a lot of time working with Pacific Biosciences, testing this on things where we knew the answer. Then we started on things where we didn't know the answer. And the bottom line is, if you look at the characterization of DNA methyltransferases, going along the bottom, this is just for some that we call type 1 and type 3, you can see there's just been an exponential growth in the last few years of the number of characterized methyltransferases that are out there. And this is quite interesting for a number of reasons. One is these methyltransferases, for the most part of restriction systems. And so if you want to then work with the organism that contains them, you have to overcome these restriction systems. And it helps if you know what they are. So one of the things that we've learned is that there are an awful lot of solitary methylases that are present in bacteria, things that are not part of restriction systems, and we don't know what they do. In many cases, we think they're involved in epigenetic phenomena, that is, they're controlling genes in ways that we don't yet understand, but we really don't know. There's a great deal more variability in the recognition sequences than had been expected. Sometimes the methylase recognizes exactly the same sequence as the restriction enzyme, but sometimes it doesn't. And if you start to think about it, let's say I've got an enzyme that recognizes GAATTC. I can protect against that sequence with something that methylates one of the A residues. But let's say it recognizes just AATT, that will protect as well as something that recognizes GAATTC. So why should the specificity be the same? It suggests that there's some biological material, some biological phenomenon going on. And one of the most interesting possibilities is that it's actually the methylase that is being selected for. So it is the recognition sequence of the methylase, and the restriction enzyme is just there to keep it honest, to make sure that it doesn't vary its specificity. Also, a lot of bacteriophages that go into bacteria, they contain methylases to protect against restriction systems. PacBio sequencing, for me, is great because you get methylase recognition sequences. Functional annotation of methylase genes becomes very easy. You can do it automatically just from the sequence. Closing bacterial genomes is very simple because of these long reads. You don't have to work out how to put small reads together. And the accuracy of sequencing turns out to be enhanced. Puzzles and games. I think I started off telling you about this headmaster at school who got me interested in mathematical puzzles. I love puzzles, I love games, I like video games. I have all the sympathy in the world for these kids who spend their lives on the cell phones playing games. I don't particularly like the shoot-'em-up games, but anything that requires you to use your mind, that's what I love. I love word games. I also play croquet. It's how I spent a good deal of my money from the Nobel Prize, put in a croquet lawn in front of the house. On the bottom left shows I also have a sense of humor. I became Dr. December in the 1997 Stud Muffins of Science calendar, much to my wife's annoyance. She thought this was a waste of time, but in fact I got quite a lot of mileage out of it at the time. It was fun. I've also been honored in a number of ways. I was knighted by Prince Charles, as shown here. I've had some things named after me. But I've also helped to organize the Nobel laureates for a number of good causes. The first of these that was really successful involved some Bulgarian nurses who'd been jailed in Libya and accused of spreading HIV deliberately in the children's hospital in Benghazi. And it was clear from the science and analysis that had been done that they were not responsible for this, but Gaddafi was just looking for a scapegoat. Went through the court system and they were tortured, several confessed, and they were sentenced to death. And I found out about this and decided maybe there was something we as Nobel laureates could do about it. I organized a campaign from the Nobel laureates. We got more than 100 Nobel laureates all signed on. We sent a letter to Gaddafi saying, this is wrong, these nurses are innocent, they were not responsible for this. I took that to the ambassador to the UN for Libya down here in New York City, And 2 weeks after, I got a call from Gaddafi's son who said, why don't you come over to Libya and talk to us about this? I went over, and after talking to him, he said, we're going to let them go. And so they were let out of prison. So I realized at this point that having a Nobel Prize means a lot of people listen to me and listen to Nobel Prize, who never would have done before. You know, my wife used to think she still thinks I talk rubbish, but students listen and so on. Anyway, so I've been involved in quite a lot there. The latest cause that I'm involved in is a pro-GMO campaign. So many of you probably know what genetically modified organisms are. I suspect there's at least one person here who would not eat them and would not buy them, even though maybe they don't know what they are, just that they're dangerous because the advertising against them has been so powerful. In fact, GMO technology is probably safer than traditional plant breeding. Not been a single accident in the 30 years since it's been used. Billions of cattle have eaten them, pigs, chickens, and so on. And a very large number of people. In fact, if any of you eat papayas, they come from Hawaii, and they are pretty much all GMO now. And so we've been trying to get word out to Greenpeace and to people, policy leaders in companies about this. We have 141 Nobel laureates at the moment. And basically all we're doing is telling Greenpeace, they claim to be a science-based organization. The science says these things are safe. Just admit this is an issue you got wrong and get on and do all the good stuff that you're able to do. If you want to find out more about this, there's a website there called supportprecisionagriculture.org where you can look and read. There's a lot of factual information. There are lectures that I've given about it. And also you can join us if you find the arguments convincing. I would encourage you to do that. And I told you I wanted to tell you a little bit about luck. So I first found out about this when I was a child at school. When I was 17, I got bored with school. We were just sort of going over stuff that I already knew, and I stopped going. And I used to go down to the local YMCA, and I would play billiards and snooker, and I became very good at this, to a point where I really thought that I was going to be thrown out of school, and I went to audition to become a professional snooker player. Fortunately, the headmaster didn't throw me out of school, and so I never found out whether I could have made it there, but I always feel there was an opportunity missed there. I might have become really famous. Anyway, I was playing in a tournament one day, and I was trying to pot something, and I didn't pot what I was going for, but I made a winning shot anyway. And then the next shot, I was kind of careless and didn't really pay attention. And afterwards, an old gentleman came up to me, and he said, if you're going to be successful in life, you have to learn to take advantage of luck. Everybody has luck, and if you don't take advantage of it, if you just feel guilty about it, you're a fool. Because everybody has it, and the guy down the hall, when he has it, he's going to take advantage of it. So always take advantage of luck. And I've tried to stick with that throughout my life, and it actually has played quite an important part. And I would encourage you, from time to time, just look back and think, what were the lucky things that directed you this way or that way or the other? So for instance, Kazuo Kurosawa here was a postdoc. I had no idea I was going to be in a lab with him. And he was a wonderful mentor. I went to Harvard instead of Wisconsin. I told you about that. Looking for eukaryotic promoters in adenovirus. This was a fairly bread and butter project, but it had the beauty that it gave us a result that looked as though the experiment had failed, but it was just that nature was trying to tell us something. And so if you're an experimental scientist, you really want this to happen a lot. So you want to be doing an experiment, and it doesn't work, not because you messed up, but because nature is trying to tell you something. I think the same thing probably applies in the world of AI. You want things that you think should work, not to work occasionally, because failure is a great thing. I love it when students come to me and say, hey, I did what you told me to do, and it failed, it didn't work. And sometimes they'll go back and repeat it, and they make a discovery. Also, many of you probably have heard of the human bomber. This was the guy, he'd been at MIT and then at Chicago. He's a mathematician and slightly deranged, and he started sending, he was a Luddite, he basically started sending bombs around. I got one of 5 letters that he sent out some years just before he was caught. One of those letters had a bomb in it and 4 didn't. I got one that was not a bomb. About 6 months after that, they found him, the FBI found him and arrested him, and he's now in prison. Taking an early plane, you probably coming from New York all know about 9/11. I was booked on that flight until 2 weeks before it took off. I was going to a meeting in California, and the meeting got moved forward one day because I also had to go to a NASA meeting. And so I flew on that plane the day before it went in to 9/11, but I was booked on it originally. Now, if that's not luck, I don't know what is. And finally, studying bacterial restriction modification systems, I really had no idea that this was what I was going to do when we first started looking at them. They've been fascinating, they've been wonderful, and they've led to an incredibly productive career. You know, I published 15 papers the year before last, I published 10 papers last year, all because of the work in this particular area. And this has been the most productive period of my life from that point, from a publication point of view. So I'd like to acknowledge the people who really helped me with this. Richard Gelinas was the guy who did the original experiments to discover splicing. Louise Chow, Tom Broker did the electron microscopy that really, because it produced a picture, convinced everybody that it was all correct. Saida Zain was a postdoc in my lab, and she made some important contributions looking at adeno-SV40 hybrids, and Dan Klessig was a student with Ray Jesslin who also did it. And then just many lab members, colleagues, collaborators, people who over the 50 years have been tremendously helpful, because I think none of us can do this stuff alone. We always have students, we have collaborators, we depend on them. These are the people who really make everything work out. And finally, my assistant, my longtime assistant, Karen Otto, made all the slides that you saw here today. So I thank you all very much. I'm happy to take any questions. Okay, well, so, you know, we can sequence genomes. We sequence a human genome, we're sequencing thousands of human genomes. And yet our ability to understand that sequence and to know how it affects human health is not that developed. We have a long way to go. It is now so easy to sequence DNA that almost everything that we're going to know about biology is going to come from interpreting DNA sequence. Our tools for doing that are not that good. This gold standard protein database I talked about is an essential part of it. We want to know what the genes are doing. There is a massive opportunity here for some work that will, I think, push the field forward in a major way, and AI methods can certainly help in this. There's just no two ways about it. We're accumulating data at the most impressive rate at the moment. In China, they're talking about sequencing 1 million human genomes. That's quite a challenge to deal with that amount of data, to understand what it all means. And if you don't have a good standard protein, and you know that this is what this gene does, how are you ever going to be able to interpret all of this? So I see the bioinformatics challenges as being just absolutely key to our ability to understand biology. Absolutely key. And I think if you look in almost every department now where you've got molecular biologists, they all get training now in bioinformatics because they can't do their work without it. It's a tool that they need. But there are less people developing the new tools that we need in this area. And so I think if you can find people who have an interest in biology but have a good background in computer science, many, many possibilities to make progress. But you have to learn some biology too, and you have to be prepared to talk to biologists and share ideas. Sometimes biologists have good ideas for algorithms. Sometimes computer scientists have good ideas for projects that biologists can get involved in.
May I ask also, in terms of the databases, how trustworthy those are? So, for example, how much errors are in these databases or mistakes?
Yeah, well, that's a good question. The answer is we usually don't know how much error is there. We know that there are some, but I think what's really important is how much significant error is there. So you expect to find variations. If I grow a bacterium, every cell on a petri dish will be mutant. It will have a mutation in one of the genes. But, sequence 8, 10, 20 of these, then those mutations disappear. Their significance is not so important. And so I think there are many places where errors are not going to be a problem, but there are plenty of places where they can be a problem. And one of the problems with the sequencing technique that Illumina developed is there are systematic errors that are made. One of the nice things about PacBio is all of the errors are random, and so if you get good coverage, then the errors disappear and it becomes highly accurate. I think there's every reason to think that over the next few years there will be even better ways of sequencing DNA coming along, and hopefully they too will be much lower in terms of the percentage of errors that come along. So provided you're aware that they can occur, for the most part they don't cause big problems.
I see.
Yeah.
Besides so I guess you all have a lot of experience in chemistry side as well. So besides the sequence, what families of signals aren't we looking at right now? Just as a very naive example, maybe location or proximity or something like that. There was methylation that came into place. What other things you think we're missing?
Okay, so think about split genes. So the exons, the bits that code for the protein, we know pretty much what they do because they're coding for protein. In between are big expanses, things we call introns. We know in some cases there are controlling elements in there. So RNAs are made and they have a function remotely on other genes. Vast majority, we don't know what they do. Big problem there. A lot of people are looking at this at the moment. Progress is, I would say, is pretty slow. We don't know too much about that. Another big area where it's clear there's a lot of important stuff is something called glycosylation. So after a protein is made in a cell, it gets glucose residues and mannose residues and other sugar residues put onto it. We know in eukaryotes, these glycosylation events are very important. If you're making drugs, for instance, they can be very important. We don't know a lot about that. There's a huge area there, and tools are needed. We don't have all the tools that we should have at the moment to study that. But they can make a great deal of difference to how this protein will behave when it's associated with a cell. We can talk some more about that. There's a lot of other stuff. Yeah.
So I just thank you so much for being here. I have a quick question is that, because I know that you transitioned from chemistry to molecular biology. So I'm just wondering, like, when you did this transition, what's your method of catching up? Because compared to your peers at the time,
right.
I guess you were probably slow in understanding the literature, understanding the experiments. And I was also switching a field actually from a legal career to electrical engineering. So I was just wondering, like, how do you apply this transition?
Right. Well, I tend to be fairly obnoxious about asking people questions. So I used to go, when I first became a postdoc, I would go to seminars and discover all the people were using acronyms. I didn't really know what DNA stood for. I didn't know what RNA stood for. And when people started using acronyms, I would tell them, I don't understand the acronym. Can you explain that? And of course, because everybody else knew what they all meant, it made me a little obnoxious. So that was one way. I would always ask people if they were trying to explain something and I didn't understand. I wouldn't pretend that I did understand what they were saying if I really didn't. And the other was I did a lot of reading. And of course, back in 1969, when I made this transition, the literature was not as expansive as it is today. But on the other hand, the textbooks that you have today and some of the review articles you can find are much better than anything we had in '69. And so it's sort of a mix. So I've always been a great believer in reading. I love reading. I like to delve into new areas. I like to read about stuff that I don't know very much about, because you just never know. Someone's solving a problem, and they're solving a problem that seems totally unrelated to anything you do, and then you suddenly realize the method they're using, well, I could use that method for the problem that I'm interested in. And I think this idea of sort of sticking with a subject and only reading in your subject is hopeless. It's not the way to make progress. It's not good. And yet, quite a lot of schools, they sort of try to get the students to focus on this, and they don't encourage the breadth of reading that I think they should.
Let's thank the speaker again.
Okay. Thanks.
A small gift for you, so you will remember.
Well, thank you. Okay.